The following program is part of a college course. These telecourses allow you to earn college credits at home. For more information, contact a college or university near you.
The magnificent diversity of life depends on the genetic instructions locked in DNA. But while DNA may hold nature's plans, another set of molecules must carry them out.
It's often said that DNA is the blueprint of life. And using that same metaphor, I like to think that proteins perhaps are the bricks and the mortar of a house. In fact, most of the things that get done in the house that we know as the cell are done by proteins.
Through time, life has produced an astounding variety of organisms, yet they all have something in common. Each living cell contains the DNA blueprint which defines their different forms and functions. DNA holds the designs of nature, but we rely on proteins to bring those designs to life.
You could understand the genome completely. You could have every single nucleotide on the genome stored in the computer, but you couldn't make a cell because you have to have the proteins to do something to the DNA to continue the life. So in that sense, they're at the very heart of life.
Proteins perform or influence every important function of life. Structural proteins build tissue. Enzymes regulate metabolism. Hormones help coordinate organ function. Dr. James Lake is exploring how different species translate the information of DNA into these life-giving proteins. For all organisms, the process begins with the transcription of their genetic code.
All the genetic information in a cell is contained in DNA. The problem is that DNA is a precious molecule, and it has to be copied so that we don't risk affecting the master copy.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
All living organisms use messenger RNA to transfer the information locked in their genes. This holds true even for prokaryotic bacteria, living in nature's most exotic extremes.
There's one particular group that we call the eocytes that are a group of extremely thermophilic bacteria that eat sulfur, typically, breathe hydrogen, and can live at boiling temperatures. They're so unusual, my wife calls them machocytes. But they are telling us things we think about where eukaryotes came from.
The eukaryotic cells of complex organisms take many forms and produce a colossal array of different proteins. On the other hand, prokaryotic cells, what we know as bacteria, need fewer proteins to pursue their simpler lifestyle. Though vastly different in form and function, both use the same cast of characters in the final stage of protein synthesis, called translation.
Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.
Practically everything that gets done in a cell gets done by proteins. And so translation is the process of taking the genetic blueprint and making proteins.
We can now read the blueprint of life contained in DNA. Using standardized techniques to isolate specific DNA sequences, scientists can translate genetic codes into computer models of the resulting proteins. Using this procedure, Dr. Lake and his team have revealed that human and bacteria cells may be more alike than previously imagined.
In the research in our lab, we're studying several molecules. We're studying a molecule which is called elongation factor TU in bacteria. It's called elongation factor 1-alpha in eukaryotes. Different names, but it's the same molecule. And in fact, when you look at the 3-dimensional structure of this molecule from a bacterium, you can almost superimpose that molecule on the eukaryotic structure. So they are doing the same functions and they have the same structure. Of course, I can't quite make out the phosphates. Can you find them here?
Sure, the phosphate is right.
The fact that certain processes are so similar in eukaryotes and prokaryotes imply, to me at least, that probably life has found some sort of solution which works very well. It may not be the best way to do things, but it works well. And once you find a good way to do something, life just keeps persisting down that path. And now we have eukaryotes, prokaryotes, everything doing protein synthesis in pretty much the same way.
Proteins play a critical role in the game of life. Whether in muscle or blood, enzymes or antibodies, these molecules carry out the intricate game plan of our DNA. The human body produces many thousands of these life-giving proteins every minute. They form a vast biochemical web so crucial, even a small mistake can produce dangerous results.
Now at the 5-day forecast.
Christopher Nance is a television weatherman and fills a busy schedule with remote broadcasts and personal appearances. His energy conceals the fact that he also suffers from a deadly genetic disorder, one that has threatened his life from a very early age.
And I overheard a physician telling my mom that because of my disease that I would probably not live to see my teens. And then, you know, I didn't really talk to my mom about it for a while. I just went into this kind of depression where it's like, well, I'm not going to live to see my teens. Okay, let's see, we can rule out marriage, we can rule out career, we can rule out kids, we can rule out a lot of stuff. Why study, why go to school?
Doctors found the problem in Christopher's blood. Normal red blood cells produce hemoglobin A, a protein which carries oxygen throughout the body. But in thousands like Christopher, the molecule is different. It behaves abnormally, causing blood cells to distort. Doctors call the condition sickle cell disease, and its dangerous symptoms are still not fully understood.
We want to answer the question, what is it that makes the vascular obstruction in this disease? And the change in the cell shape, the change in the characteristics of the cell membrane, the change in the physical properties of the cell, because that's the key feature in the disease process.
The flexibility of normal blood cells allows them to squeeze through even the narrowest blood vessels with ease. Sickle cells, on the other hand, are more rigid. They rupture easily and cause obstructions, which are both dangerous and painful.
Think of your body as a pin cushion, but instead of you pushing the pins into the cushion, the pins are on the inside trying to push out. And that hurts. In your fingers, your joints, your neck, your lungs, you take a deep breath and, oh man, it's like the pin's expanding out of your chest. Very, very painful.
Christopher's pain indicates serious tissue damage that can shorten his life. Doctors trace this threat back to an error in Christopher's genetic code for making the protein hemoglobin.
You start with this genetic defect, all right, at the DNA level that instructs the red blood cells to make hemoglobin S instead of hemoglobin A. Hemoglobin S, because of the amino acids sequence, undergoes a conformational change, a change in its structure.
Genetic defects occur when the nucleotide sequence of DNA changes or mutates. In the case of sickle cell disease, a specific base in the code for hemoglobin is changed from thymine to adenine. Molecules of mRNA then translate this mutational change into a different protein with a single amino acid substitution that has devastating consequences.
And because of this change, hemoglobin S has the property of polymerizing under certain conditions. And by polymerizing, I mean that it will form a crystalline structure. The crystal structure resembles a straw. That is, it's a microtubular structure. So it has a hollow center, so to speak, and a spiral of hemoglobin molecules going around and around. So it forms this long straw-like thing. And these polymerized hemoglobin S crystals then distort the red cell from its round biconcave shape, which is very flexible, into a distorted half-moon shape, which is very inflexible.
Research also indicates that sickle cells may stimulate other body cells to express a sticky protein, which aggravates the condition. In other words, the disease may produce blood cells which are both rigid and sticky, a deadly combination.
So we have this population of relatively rigid cells passing through the bloodstream, passing through these very small blood vessels, and if a cell is temporarily held up in that process, then the very rigid cells can't get through an aperture that's suddenly more narrow. And as we were discussing, this is sort of like what happens in the freeway when somebody breaks down in the second lane and people are trying to get by, and it becomes more and more difficult to get by, and eventually things become pretty much slowed down to a stop.
In the body, these vascular obstructions can damage vital organs in a very short time, which explains why this disease claims the lives of those so young. Living beyond his teens, Christopher Nance has beaten the odds. Still, his life has not gone untouched.
Gosh, I'm now in my 40s, can you believe that? And I wonder about the damage that I've done to my body. I know that through the hundreds of crises that I've had, that I've done irrepressible damage to my lungs, to my heart, to my liver. I mean, I don't have a spleen. That was eaten away, as it was described to me by my doctor, by a crisis I'd had. And you just hope that it doesn't affect the vital organs that you need.
How many of you guys are still reading? Good.
I guess the devotion that I have to the school kids has a lot to do with the fact that sickle cell disease is a childhood killer. When you realize that there are children that will never reach their potential, never know what they want to be when they grow up, and never become it, then you feel that there's a great injustice in the world. And whenever you see an injustice, your first reaction is, I'm going to correct it.
Christopher has dedicated himself to helping doctors find a cure for sickle cell. However, the means to alter the genetic code which causes the disease remains just out of reach.
We would like to either instruct the genes you already have to produce, not to produce hemoglobin S, but to produce one of the other hemoglobins, or give you something that would replace the hemoglobin S. Not an impossible task, but it's going to take some time before this is feasible.
A translucent glob slowly transforms itself. A slime mold is on the move. This curious organism reveals how cells control their protein production and may shed light on the mystery of human diseases such as cancer. From molds to maples, all living organisms use the same process to translate the genetic information in their cells. However, different cells produce different proteins in response to a variety of control mechanisms. For example, the complex eukaryotic cells found in both slime molds and humans require a network of interacting control systems. Prokaryotes, on the other hand, use a much simpler mechanism called an operon, a group of related genes controlled by a single on-off switch.
A prokaryotic cell, in fact, responds to a much more simple environmental system. It needs essentially to grow, and it needs to respond to what food is in the environment. Operons allow the prokaryotic cell to respond to these relatively simple changes. Eukaryotic cells are in most cases much more complex. While it has relatively simple pathways that are used for metabolism, most of the complex developmental or program that's in a eukaryotic cell is to allow it to differentiate into one cell type versus another.
As complex organisms develop, their cells specialize or differentiate to perform specific functions. Some carry oxygen, some move limbs. Others store fat or send nerve impulses to the brain.
Cell differentiation is the process by which an individual cell will alter its developmental program. For example, you might have a precursor cell that would then differentiate into possibly a white blood cell or a red blood cell. The process by which that stem cell or a precursor cell differentiates through that pathway would be considered the process of cell differentiation. My laboratory's particular research interest is in understanding both the general process of differentiation and understanding what types of mechanisms eukaryotic cells use to control differentiation.
Every living cell holds all the genetic codes required to build an entire organism. But as each cell differentiates, it uses only some of those genes to produce specific proteins. These proteins dictate each cell's destiny. Using cellular slime mold, Dr. Richard Fertel hopes to explain this complex process known as differential gene expression.
The goal in understanding differential gene expression is trying to understand or elaborate the actual mechanisms by which each of these genes is used in various cells. For example, hemoglobin, we know, or globin genes, are expressed only in red blood cells. What are the mechanisms by which these genes are turned on only in red blood cells? And what are the mechanisms by which these genes are kept from being expressed in other cells? In order to understand differentiation, and therefore to understand various disease processes, we must be able to understand how many of the genes in the human body are controlled.
Enter dictyostelium, the cellular slime mold. This organism may hold clues to the relationship between cell differentiation and protein expression. Scientists have identified some internal biochemical cues which are analogous to hormones, long recognized as triggers for differentiation in animals. But their work has also uncovered the influence of external factors in this organism's extraordinary life cycle.
In dictyostelium, it's known that environmental cues such as light is able to trigger the final differentiation state of the organism. So dictyostelium in the wild lives in deciduous forests and eats bacteria on decaying leaves. After it aggregates and forms a multicellular organism, it then forms a stage which is called a migrating slug, and it basically looks like a small tobacco tube that is capable of migrating along the forest floor. When the organism then sees overhead light, it then changes from this migrating slug to form the mature fruiting body. And the environmental reason for this is that if you're a dictyostelium, you do not want to form a mature fruiting body underneath the leaf because you will not be able to disperse your spores.
As alien as it may seem, this organism offers researchers a valuable model that may help reveal specific pathways of gene expression in humans.
By defining the pathways in a simple system such as dictyostelium, we can now go back and relate how the pathways may be perturbed in mammalian cells. That is, how a particular mutation in a gene in human cells causes disease, and while the unmutated gene is perfectly normal.
Slime mold may hold the key to some perplexing human problems. For example, when dictyostelium cells begin their lives as free-roaming individuals, they contain small amounts of a protein called NM23. But when the individual cells collect into a single organism, the concentration of NM23 rises dramatically. Scientists have observed a similar concentration of NM23 in human cancer cells.
The NM23 gene was shown in certain types of cancers to be potentially important for maintaining the metastatic state. That is the state at which cancer cells now move in an unregulated form. In dictyostelium, a similar gene was identified and has been shown to be implicated in controlling a number of pathways that are involved in the aggregation formation in dictyostelium. The aggregation formation is the stage in which a large number, up to 100,000 individual dictyostelium cells, form a single multicellular organism that then undergoes differentiation.
Dictyostelium may be pointing the way to important insights about the deadly behavior of cancer cells. More importantly, this simple organism sheds light on the crucial role proteins play in the living world.
The reality is pathways are complex, differentiation is complex, and if we're to understand it, we must not only understand each of the individual simple pathways, but we must understand how these individual pathways are put together to make a complex organism.
We've been able to compare the protein sequences that are found in bacteria and the protein sequences that are found in animals and in humans and in plants. We find out that there are common sequences. In some cases, they may have 50% identity between bacteria and humans. So it's really becoming quite clear to people that all life is related.
If you looked at the protein for skin, for example, and compared that to the protein for fingernails, the difference is a few sulfur molecules. And that makes it soft and flexible, or makes it hard and clear. It's simple and elegant in many ways, and unbelievably complex in other ways.
All living organisms depend on proteins to survive. In their myriad forms, they perform the work of life, yet we all produce them in the same way, from the transcription and translation of our genetic codes. These complex mechanisms carry out nature's intricate plans in the ongoing cycle of life.